Hi, everyone. It's really a pleasure to start this year's IICD seminar series with our guest today, Professor Fabian Theis. So Fabian is really well known as a leader in the computational biology field. He's the director of the Helmholtz Munich Computational Health Center and the scientific director of Helmholtz AI, and a full professor at the Technical University of Munich. He's also holding the chair of mathematical modeling of biological systems and associate faculty at Wellcome Trust Sanger Institute. Through his training, he studied math and physics, has PhDs in physics and computer science. He was a Bernstein Fellow at the Max Planck Institute, and in 2007, he became a group leader at Helmholtz, an associate professor at the Technical University of Munich. He has many prestigious awards and honors. The most recent one is receiving the Gottfried Wilhelm Leibniz Prize in 2023 by the German Science Foundation. And Fabian is really a pioneer, as everyone here knows, in developing machine learning methods for analyzing single-cell data, data integration, modeling cell fate, lineages, dynamics, as well as software infrastructure. I think everyone here is used either at least Scampi or a couple of methods developed by Fabian's group. And we're really delighted, looking forward to hearing his talk today on generative AI for modeling single cell state and response. Fabian.
Thanks so much, Elam, for the kind invite. It's really unfortunate I couldn't come in person. It's always just such a blast being in New York, and I hope we can catch up a bit later. It's just family-wise. It's a bit reserved. So thank you so much for coming. I'm sort of take it easy a bit in the beginning and show a few things that we're doing, and hopefully in the end going a bit more towards this sort of new ideas on foundation models and perturbation modeling. I'm very happy to sort of get some feedback, think where you're going, also at the end highlighting a bit on the spatial transcriptomics side of things. But for those of you sort of maybe also not so much in the single cell side, so let me maybe take the intro to a bit laid back and let me just start sort of with the general setting, right? I mean, if you want a sort of very high level, I guess in AI machine learning, where we try to learn from data, and traditionally we've been having this sort of discriminative models, always trying to look for some type of supervised setting, and this has been really revolutionary, right? So let's say 10 years ago or something like that, when AlexNet came out, we solved this supervised problem of image classification, which hadn't been. When there's a new sample coming, right, you have the classifier and you can do classification. So you can do cats versus dogs images, which is seemingly simple, but, has been really bothering computer science for, I guess, 50 or 60 years before, these seemingly trivial tasks that every, 2-year-old kid can do was actually be able to be done by a computer. And now, of course, we have all of these pre-trained models and can do this robustly. And I think that we all, of course, appreciate this revolution of deep learning that has been enabling many of these things. And this is 10 or something like years ago. In comp bio, we're in parts using deep learning for images and stuff, for others maybe not. And I think it's quite interesting when some of these sort of large-scale embedding techniques come along, in particular given the data deluge that we're currently facing. So if you want to go back to this classification scheme, right, I think particularly unsupervised methods or self-supervised learning has been taken, I think, the recent years in machine learning by storm. But in many of our cases, this has been around and been needed for a long time. There you don't have labels, and you want to sort of group cells, or case, or objects into categories. You want to cluster them. You want to find a low-dimensional representation. You want to find what makes up, the core structure of the data. Potentially using that to build your models, your system's biology of the particular system. The type of data we are looking at, and we're very much interested in, at least initially, is single-cell genomics, where you have these beautiful techniques, for example, based on microfluidics, where you can pick single cells, embed them in these little water droplets embedded in oil that work as these little mini Petri dishes. You can then add labels to these particular cell labels, then sort of fuse everything together and use a bulk RNA-seq experiment, and thereby have these single-cell resolved expression profiles. So essentially what you get cellular state for many cells. And this is really becoming big data. With current experiments, we easily get hundreds of thousands of these transcriptomes. And for the older ones amongst us, remember when we started with n equals 3 microarrays, now we have these millions of high-dimensional profiles around. I think it's really a fun area. And one thing, of course, you can do these unsupervised things where you have your dots, which would be gene expression values, but not in 2D, but in 25,000 dimensions or something like that. You can then ask again for groups. And if you ask for groups, for cells that have similar expression profiles, you, for example, get cell types, right? That would be one of the typical approaches that most of the single cell analysis workflows initially do, grouping things into categories and then, arranging these. My lab has been sort of going beyond that clustering, and it's not my lab, a lot of us have been having this type of interest. Essentially, for example, here looking into a three-dimensional projection of high-dimensional single-cell RNA-seq state, in this case from collaboration with the Heiko Lickert lab here from Helmholtz, from samples in the intestine, where you see they don't form just clusters, but there's all these connections. You see how these clusters are connected. In this case, for example, this cluster over there would be intestinal stem cells that would connect to these endocrine cells here via this bridge, as you might have seen from flying through. And this bridge would be, for example, enteroendocrine progenitors that prime these cells before they become these differentiated cells. And for example, Dana, I believe Elham, you might have been working with her, right? She has been quite famous for a Wanderlust algorithm. And we've been, as well as many others, following up, for example, us developing diffusions, using essentially trajectories in this initially K and N reduced space, took for Markov chains to then learn these type of transitions. And there's a lot of fun to be had, a lot of methods for these pseudo-temporal ordering, branching methods, and so on. And quite interestingly, what we did here, so you might not care about cells, but I added this one slide because I think whatever methods are developed there, potentially could be also generalized in other settings. You might, for example, care in this case, we've been using quite similar techniques for a patient classification. In this case, disease called diabetic retinopathy from eye backgrounds. We just arranged them, and we could also sort of learn trajectories of potential disease progress, and then have been coupling this also with actual longitudinal measurements. Won't be speaking about that, but I think it's an interesting area to keep in mind that, going beyond just the grouping, I think is an interesting idea. So let's get back. So once we have these groups, we can really start doing things. I think one of the biggest endeavors in the field at the moment is the Human Cell Atlas, hugely international consortium or collaboration entity with, I think, more than 3,000 researchers all around the world, set up by Aviv Regev and Sarah Teichmann, but many of us contributing. I'm also contributing in the OC, essentially trying to build this periodic system of cell types, if you want. So just giving us categories for the different organs, for example, indicated here in this nice review from Sarah. We've also, having very many type of disease settings that you can project on. So I think this could develop into quite exciting results for all kinds of downstream analysis. I'm saying this because I think actually coming up with the idea what such an atlas is for a particular site is not that trivial, actually involves some type of machine learning. So the first question I want to ask in this talk is what actually makes an atlas an atlas? So we talk about Human Cell Atlas, and already first papers, for example, here for single cell data in the lung, something like 4 or 5 years ago, coming up, they already call this thing atlas because it's such a beautiful metaphor, right? We take samples, in this case, I think from a few patients, and within one lab, and we put them together, and it gives us some description of the variation in this thing. But many other papers have been coming out then, and question is, if 50 different labs make similar things, what's the actual atlas? What's like the shared structure? So one question we ask is, can we somehow unify this? So this idea of building a cell atlas can be actually reformulated into a machine learning question, namely of learning an atlas. And this is sort of this data integration thing that Elham was already hinting at. Here I have another metaphor of actually looking at an atlas of the world. This guy, Zach Sieber from Red has actually putting together, asking around campus people to draw a map of the world. And you see, there's like these different sort of things being drawn. I guess I might forget Australia or Madagascar or something. But if you put all of these together, and he's been sort of doing this by hand, just superimposing, you kind of see that, on average, crowd intelligence does not forget Australia. This looks a bit strange, but that's kind of what you get. So similarly, what we would be doing in our single cell data sets is like if you go into one lab, one of these papers, we get our nice cluster and everything is fine. But if you put all of these together, it looks like that. So it's kind of a mess. So you have sort of a lot of batch effects actually dominating the major axis of variation. In this case, I've been plotting this on a UMAP just for illustration, but of course, this is just a sketch. But similarly, you can imagine that you need to do some type of data integration. And typically, it's a non-rigid, a non-linear superposition of these things to actually then get cell types to be roughly overlapping, but of course, to still keep biological variation. That's a typical trade-off. Quite a few techniques have been developed for this. For example, also from Raoul Satij's lab from New York Genome Center, to come up with some of these ideas. He's been actually pioneering quite some exciting things here in his Seurat toolbox. We've been coming up with some metrics for measuring these and being able to compare this. And essentially, this has been leading us towards more robust integration. I want to speak about machine learning, right? So I wanted to show one class of methods that has been becoming really popular. This is based on an unsupervised learning technique called an autoencoder. Sure, many of you know this, but let me just walk those through that haven't seen this maybe so much. Essentially, we take, for example, a gene times cell matrix. So we have a number of genes, and we just feed in many cells. What you do is, squeeze them down towards a lower dimension representation. This is called a bottleneck or latent space. Then you blow them up again. Remember, you don't have samples. You don't do a classification, something like that. So you make the output as similar as possible to the input. That's a loss. Typically, if you take mean squared error, everything was linear, you would be just learning in PCA. But in our case, we do something more complex. We adapt the noise here. You do nonlinearities. So you get sort of low dimension representation here. And of course, you also reconstruct. Remember, you feed this through this bottleneck. So you just remove away all these sort of noisy, sparse observations, and you try to learn the key process. In this case, for example, sort of some type of differentiation process. And interestingly, you can do something called a conditional autoencoder, conditional variational autoencoder. We have a sampling version here as well, of course. That's a very traditional thing in the field. So if you take batch information here, which would be saying, this comes from lab 1 versus lab 2, lab 3, this thing during learning can adjust for this and sort of feed in particular pushes for one batch that are different from another, thereby sort of giving a more efficient way to encode the bottleneck layer, removing batch information, because that sort of gives you the most efficient representation of the data. So this thing should be learning a more robust batch representation, as we saw before. And indeed, this is the case. This is sort of what happens in sketches. And this is actually what happens in reality. For example, in this case, we've been really asking for two-dimensional bottleneck. In many cases, it's typically high-dimensional. But here, just two-dimensional of PBMCs colored by cell type labels. And you see this two-dimensional neuron 1 versus neuron 2 looks like typical UMAPs that we see. So our first paper, Nir Yosef and my lab were the first to propose autoencoders in this setting. And initially, I've been sort of just mostly talking in my lab about denoising. Remember, Elham, this was when Dana had magic and so on around. But then when I saw this and saw, oh man, this bottleneck is actually doing something interesting, because now you have a parameterized version of learning the latent space. This is when a lot of us got interested in this latent space here. And this is now what's happening ever since. A lot of people now adding priors to latent spaces, making things multimodal, and really trying to reformulate the biological questions in terms of constraints in this thing here. And just sort of to say that this becomes popular, initially, a few people of us have been doing this, but we've been running a few competitions, I think 3 now at NeurIPS. First one, we had 300 people. The next 2, I think something around 1,500 people participating at each of these NeurIPS workshops. And I think pretty much all of these winning methods have been using one way or another of such a representation learning technique. Initially, also autoencoders, now more transformers and so on. So let's try to use these techniques, this sort of sketch that I showed you before. We have some variation of that, and we'll be discussing this a bit later, to now learn an integrated lung cell atlas. This is work by Lisa, a very talented PhD, who has been actually a intern at Dana's place or also before. I'm very happy that she joined as PhD, and Malte, who now moved on, to become a PI in a really exciting collaboration with the essentially whole human lung cell atlas network, but particularly Sasha and Martin, where we've been initially taking just 14 data sets, harmonized and annotated something like 500,000 of lung cells, and harmonized also the metadata and anatomical location, and then combined all of this into this representation, okay? So remove batch effects, and here we found this type of map. And because now, I want to call it an atlas, I make it a bit more visual by adding a circle here. So now you get the thing, right? So you have here your continents of, let's call them epithelial cells, immune cells, as well as endothelial and stroma cells. And this is now homogenized, if you want, across the different sources. So you can actually zoom in and you get like all your annotation for the epithelial, endothelial, stroma, as well as immune cells. And this has been really just made possible by really expert annotation in many different levels. So this was a lot of work with experimental partners. So now that you have this, you can now ask different things, right? And for example, what we have been doing, we've been now extending this and also adding disease on top of this. And you can, for example, ask, is there one particular cluster that's sort of shared with different disease phenotypes. In this case, for example, we had a bunch of them that have some pulmonary aspects, some also carcinoma and other diseases as well as COVID. And we found that one of them had this SPP1 high signature in monocyte-derived macrophages. And this is apparently relevant for scar tissue formation. So you can sort of look at shared patterns of disease in this sort of integrated thing across diseases. So I think it can be really exciting resource for projecting your own data on top of this. And this is actually what you want to do. I talk about projecting in a minute because that's a fun machine learning question again. But I just want to say we started with the lung, but of course, many in the field, us included, have been generalizing this for other settings. For example, here, our center here in Munich, Helmholtz Munich, is focusing very much on metabolic disease in addition to airway diseases. So with Karen, we've been doing a mouse islet atlas that came out earlier last year. We've been integrating a bunch of mouse data sets, particularly, but also projecting human on top of this, and then being able to ask how, for example, differences between various mouse models would actually reflect on top of that sort of integrated map. Are some of them similar, some different? How much are they realistic with respect to a human setting? And you can ask questions on these atlas, and it's also a really interesting resource. Similarly, we've been doing this for organoids, which I think is exciting because there's more and more data coming out, but it's not really clear how much they reflect versus the actual human setting. So together with Bart Treutleins lab in Basel, together with Greg Camps as well, we've been building a human neural organoid atlas as well as an derived organoid atlas, both of them out on bioRxiv, and I think a really exciting resource to play around because there's also your time structure, but also to maybe use for your own experiments. And there's many others happening at the moment within a human cell atlas for pancreas, heart, liver, skin, as well as development. So if people are interested in this, really happy to talk about this. The whole HCA is an open thing, and be really excited about people contributing to this. So to summarize this first part, referencing a cell atlas can be potentially defined as an AI-based integration. Call it AI-based, but you just saw the method for that of single cell profiles across lab. All right. So that's nice, but it is for the building. So what do you actually want to do with these things? I think the first use case that a lot of people have is they want to just essentially annotate their own data sets and not cluster them anymore, something like that. So, I think we will be really transitioning from how we do standard analysis towards these sort of using of references as we have been doing in genomics, right? Nobody of us is assembling a genome anymore, right? So to use this, essentially what we want to do, we want to take an atlas, we want to project new data on top of that. This is a technique that we have seen in computer science and machine learning for a long time. Essentially traditionally doing separate tasks, well, you don't want to do that. You want to combine them, right? So you do something called transfer learning. You take this one here and you transfer it to a new setting. And this is what we did in that work, which I explain in a minute. Essentially, you have sort of your background model and you superimpose, you project your new things on top of that, and thereby you can sort of see where new things fall, particularly where there's sort of a new cell type, something like coming up. This is based on what we call architecture searches, work by Mo, super talented PhD in the lab, who's now a PI at Sanger, and welcome to Sanger in UK, where we've been essentially adapting this autoencoder setting towards adding new conditional settings. Essentially, what we did now here is asking how can we project onto a given atlas of PBNC and lung data a new sample where we have been getting, I think, lung lavages from people in different COVID settings. This was literature data, but because now we have this nice atlas, and this was before we had the lung cell atlas, so this was basically a smaller one that we did ourselves early on without as many data sets. But now we project those COVID samples on top, and you see that, for example, here a few of them fall, and here's also there's an interesting difference between moderate as well as severe COVID. Because you have an atlas, you can now ask, hey, what's different here and what's happening here, right? You have a landmark, you know which streets are there, so to speak. So zooming in, right, we know, okay, yeah, here's actually some immune cells, and here's actually macrophages. We won't be speaking about the CD8 T cells here, but for the macrophages, we see this interesting difference. So let's zoom in. And what we find is that actually this moderate ones, they're associated with markers such as FAP-P4, which are known to sort of highlight these tissue-resident macrophages, in contrast to the severe case where these monocyte associated ones are actually originating from. All right, so that's interesting to you. So you can map on top and you can see where things fall. How can we, though, sort of do this mapping in a more interpretable fashion, not just sort of saying, hey, we kind of smoosh this on top, as Stana likes to call it. So for this, we've been coming up with an extension of our current thing, current architecture that we call SE-Poly, which we call the single cell population level integration. It's worked by Carlo and others, where the idea is now that we want to learn the integration as well as also map cell types and do condition embedding. So let me explain this in a bit more detail. So here we have a reference. So we start with an encoder. Just give me a second. Sorry for that. So we take this encoder decoder setting that you saw before, but now we add a bunch of reference landmarks, and we can, for example, ask, can we use annotated information to then also transfer to the query setting? So we just modify this, have a semi-supervised loss function. I think it's quite straightforward. But in addition, we can also now ask how we deal with these conditions that we saw before. Remember, we use these conditions to encode which the different batches are from in order to be able to integrate. So whenever you have something new, you need to sort of add conditions here. And this gets kind of bad if there's like hundreds or thousands of patients or subjects coming together, which is actually reality now in many of these data sets. Conventionally, this was one hot encoding, so this gets larger and larger. And the key idea we had here was to replace this by actually learned conditional embeddings. So you fix a space here, you give them some vectors, and instead of sort of learning the weights here and adding new weights, just have this additional dictionary, so to speak, and you learn this during the training of that network. So you have this additional space, you can see where they form. And then in query mapping, you just sort of add some new vectors here, and you sort of see, do they fall close to one or the other sample here? And for the landmark cost, you can sort of also do an additional query loss. We'll be speaking about that, but I think this is actually interesting for interpretation. So what we observe, and I won't be showing this just came out, we observe that the performance of this approach is actually competitive with respect to methods that do just this conditional one-hot encoding, but also with respect to these ones that use prior information. So it's actually a really nice trade-off between both worlds, and you can sort of use this one or not, but I think this interpretation is very useful. So what we do, if you apply this now to the lung cell atlas, we get a similarly nice embedding. But what we can now do, we actually even show that the performance is on par, maybe even a little bit better than the one that we used in the original atlas. But in particular, we also get the sample embedding. Remember these different directions that tell you where things come from. And for example, here, and now we just reduce this embedding space to two principal components. And we see that there's actually some different samples, each of these dots now not being a single cell, but being a study, where you have then superposed all of these single samples. And you see there's something different happen. And for example, here, I think this would be just study origin, as you see here. But if you highlight this one according to sample type, you see there seems to be actually something happening here with organ donors versus a live one. So you can sort of see where origins of sample variation is. And you can sort of push this a bit further, do this also for PBMC atlas. So if you're interested in large-scale PBMCs in this one, we've been actually sharing a really large-scale integrated data set across, well, atlas, if you want, across more than 25 data sets with more than 2,000 individuals, 8 million cells. That looks like that, okay? So we've been essentially just curing all cell by gene for PBMCs and putting this together. But because now we have these embeddings, right, we can see where the origins of all of these T cells actually are in terms of the 2,000 individuals. So if I plot now those 2,000s, which would be those different vectors, which we would have formulated as one-hot encodings, now live in this additional space, well, we can plot them here. And you see now each of these dots is now a person, where for each of them you have something like 5,000 cells on top, right? In order to then make up this. So it's kind of cool. You kind of do like a UMAP now, not of the single cells, but like sort of a level higher. And for those of you who have been doing original systems biology work, right, we had this dream at some point to have these multi-scale models. In a sense, this could be an approximation of that, right? You have your sort of the single cell scale, and you have sort of this population scale. You can ask them where all of this variation comes from. And you see, for example, if you just take the first two PCs, that the first one is dominated by assay origin. So you know where they come from, different versions of 10x or sequel dominates that order of variance, as well as disease is actually sort of playing a big role. We can interpret this embedding. All right, so to summarize, this gives you some this population level integration embedding, and it could potentially really help going towards more multi-scale models. And what we're actually currently doing is now adding really disease labels for each of these people. Because in the end, you want to potentially have a better interpretation of what's sort of differential versus various disease stages, for example. And this can be formulated in this unsupervised setting, but what's actually more clever here is that we extend this now. We use something called multiple instance learning, which gives labels for bags and goes here. If someone's interested in this, I'd be really happy to discuss afterwards. I think there's a lot of potential here as well. So to summarize that part, how can we use reference atlases? We just use transfer learning, and then we map essentially novel data sets towards this reference in a hopefully interpretable fashion, or somewhat interpretable fashion at least. All right. So I call the talk generative, and all of these models are generative in the sense that they are using variational autoencoders. You can sample from them if you want, but the key sort of novel thing that I think AI has been driving machine learning in the past few years, of course, is being generative about it. So not just sort of discriminating between things, but actually making distributions that make up these things, right? So you don't want to just know, hey, what's different there, but you want to sample from this high-dimensional joint distribution P of X, Y, if you have a supervised case, or just from this distribution here. And I remember like early on, we've been trying to use something like high-dimensional mixture of Gaussian models and so on. And I do think, I don't know, Adam, if you've been around, wasn't there this method called Biscuit from Dana, which I think was also doing something like that? We kind of struggled with this as well. And I think we've been all overtaken by these beautiful ideas of, for example, diffusion models and so on that really very robustly can sample these high dimensions. So being generative there, I think is exciting. And this is what you can do with these models now. I mean, isn't that crazy? Remember how bad computer vision was like just 5 years ago? Now you generate pictures of the Pope in funny jackets that are not real. So he didn't wear the jacket, right? It's completely generated. So let me take this metaphor a bit further. How can we apply this in our setting? So I've been asking Midjourney to generate now images of the Pope in white fluffy jackets. Why? Because I want to sort of take this idea to then model a cell's response to drugs. And I sort of take that in a minute, so just bear with me, okay? So I take the Pope in a white fluffy jacket, I get these type of samples, and I'm sure it can generate many more of these. And quite funnily, if you ask Midjourney to also add sunglasses, suddenly the pose of the Pope starts becoming cooler as well. So there's some interesting covariates that are not maybe entirely clear. But what I now do is I remove now this one covariate, okay? Because I want to do domain translation essentially. So I just generate images of the Pope, and they seem to look like that. So now I did this sort of operation in latent space, which in this case will be just sort of the description space of text. And now I add, so if I take this difference, what am I left with? Well, I'm kind of left with the concept of white fluffy jacket, right? So now let's add this concept of white fluffy jacket to New York Times Person of the Year 23. Who knows who she is?
Taylor Swift.
Yes, exactly, right. And this is what Midjourney thinks Taylor Swift would look like. Kind of looks real, right? With maybe interesting colors here. And what do we generate? We generate pictures of Taylor Swift in white fluffy jacket, of course. Okay, so much for that joke. But you see what I wanted to say, right? So we take this latent space arithmetics, if you want, we can generate samples. And this is kind of what we did for cells. So we started with, let's say, some perturbed cell. That could be now not the cell in the white fluffy jacket, but maybe sort of a cell seeing one particular drug or a particular CRISPR screen or something like that. And then we subtract the ground state, and we go to a new cell type, maybe a new patient, maybe a new cell line, something like that we've measured just in ground state. And then we generate samples of this perturbation. And you can do this for one, but if you do this for many perturbations at the same time, you potentially can go into this idea of virtual drug screens and so on, which I think is potentially quite exciting. So this is what we did. Our first thing was already quite some time ago, 5 years ago, so we called SEGen, worked together with Mo, where we've been in latent space essentially doing this arithmetic, so this addition and subtraction, which was a concept that we borrowed from computer vision, thereby generating new samples for others where we have seen the effect of the perturbation. That was working okay, and it's actually still quite interesting technique to look into, but we've been since then pushing this sort of going beyond these latent space constraints and so on. So this is the idea here. This is work together with Facebook and our Meta AI, in particular Anna and David from them, and as well as CodeTrap now. We've been doing some very exciting data that just came out, where we now take, again, our encoder-decoder setting, but now want to be able to sort of model this latent space arithmetics. So what's the generalization of just plus minus? Well, it's just a linear model, right? So this is what we did here. Let's say we have our perturbations. It could be drugs or something else. We have some embedding of these, and then we just, say, hey, we want to say if we maybe have sort of many of these, we can just sort of add them in a simple linear model. So this would not work by itself because you can sort of put all of the drug effect into the Z. So you need to combine this one with an adversarial loss here that tries to learn about the perturbation, and you sort of try to minimize this loss while sort of being able to reconstruct as well as possible, okay? So this is trade-off between these things. So this one does not know about the drugs. So the drug information is just added here. So during training, you would be sort of learning the effect here, and then in inference, you can see what the particular effects will be. So with this, we could potentially predict novel conditions, maybe even have a drug response model, and in particular also look at this drug latent space. So this is what we do here. This is this beautiful study from Cole called Cyplex, which has been following up with a few more things. Actually, in the paper, we have a beautiful combination study as well that Cole made. In the original one already was more than 500,000 cells across more than 4,000 perturbations. So really large scale screen. And essentially, you don't really see much happening across these 3 different cell lines, but you can now train the model. You hold out a few conditions. For example, we've been training this on low dosage drugs. We hold out prokinostat, this particular MCF cell line, and we see then that our predictions kind of match up with the true ones, in particular if you compare them versus control for all of these transcripts here. We can do this more systematically and correlated predicted versus true, and we get nice R squareds here for these holdout combinations of cell lines and drugs for particular doses. And then we can also see where this is coming from, but in particular, we can now draw this latent space of directions of the drugs. And this is what we get here, just to reduce to 2 dimensions. And you see that, so you see some structure of what these different effects would be, and there seem to be some clusters of similar effects happening. So you can sort of map out drug space. All right, so that's nice and it works. And in the paper we've been showing this also perturb-seq and genetic setting. We added interpretable priors and so on, but we have not really extended this initially already for making sense out of the drug space. So this is what we tried here. This was a paper we had at NeurIPS last year, worked with Leon in collaboration with the Gunnemann lab from TU Munich. We've been now adding also a perturbation network where we really try to learn drug structure addition. So I think because we have these flexible models, we're entering this interesting time that you can add additional priors of sort of orthogonal things, such as in our case, the perturbation setting. So we had now this molecule encoder, combined this with sort of a scalar version for the drug scale and this perturbation encoder. And we just sort of put all of this together. And suddenly we trained on the low dosage, and our chem-CPA, so this chemically informed CPA method, was able to predict better holdout on differential expressed genes of reconstructed ones. And we could show that this one was actually doing a better job for prediction, the single one, actually even out of distribution, where we went to the drugs were completely unseen, but sort of somewhere in the convex hull of the existing ones, in particular also for combinations. So I think that's an interesting idea. This got on the cover of MSB recently, and you sort of see this idea of sort of decomposing them and then sort of moving them up. If you're interested in this, check out the paper or the NeurIPS version. I think there's quite some potential for follow-up. Just to summarize this, and to model a self-response to a drug, essentially we can use some of these generative AI ideas to extrapolate response profiles from other drugs and using references. So where are we going with that? So I think this vision to do now experimental design by modeling perturbations, I think is a quite exciting and direction to go, where this space of potential experiments that we can do in biology is always much too large. We will never be able to map this out. So having in the future maybe some AI sitting on our side that would sort of tell us and give us some hints, which ones to do next, where we could potentially explore what we learned, I think could be an interesting idea. We put this forward some time ago, and I think that could be maybe going somewhere. And you could potentially also use this then for in silico experimental design, right? Let's say if you have some type of disease process happening here, from maybe control going towards these overweight mice that I showed you at the beginning, we could maybe then use generative AI in that sort of obese setting, sort of make our virtual screen, say, hey, which of these drugs would actually move us, which place in our dense landscape, maybe even of cell state. And yeah, maybe this one is doing the job. So we rank them, and then you sort of can follow up the next experiment. I think it's potentially a cool idea. And, that could maybe really go towards this foundation model. So this idea of training a model on all kind of perturbations and being able to, predict you for particular cases where we go, this is the idea of aggregating data. I'm going to be talking about this a bit more. Just wanted to say that, of course, many of us are thinking about this. There has been this very nice review or perspective from colleagues, sorry for that. Oh, I'm blanking on the name, on multimodal self-supervised pre-training, just like on all kinds of data. I can just speak about omics, but you could potentially also add other modalities, and which your particular interest for. But adding more of this and giving you a better representation, I think will be the future for many of the things we will be doing. So let me talk a bit more in the remaining, I think, Elham, 8 minutes or so, if I'm correct. Stop me if I'm going too much over time. I want to talk about this idea of foundation model, right? The idea of gather data at scale, train a model, then evaluate its performance, and just fine-tune it, particular downstream use cases. That's the idea. So you have a lot of data, just do this type of self-supervised training, and then you get your specific examples. That's like a way to go. And if you want to think about this like language models, it's like one of the best examples, right? If you want to do natural language processing, if you want a chatbot nowadays, the state of the art, you need to have in the back end a large-scale LLM that has been pre-trained in such a fashion, otherwise you won't be competitive. So think in the future, it could be also in our case that maybe there's some of these backend models that just enable our downstream use cases that we then actually care about. And of course, scaling is one of the key aspects that, all of these foundation models have been showing in various settings of NLP, particularly where, people came up with various tasks for the longest time just were sort of really badly solved. And suddenly, you see this explosion at some point of computation. And I think already 2 years or so ago, OpenAI made these predictions that, our test loss keeps going down the larger we make our models in terms of data as well as compute. So this scaling behavior, I think, is exciting. And maybe we're also entering this regime where maybe we shouldn't even play around too much with models, just sort of throwing more data at it if we have this flexible, transform model-based models. Not clear where this goes, but I think there is some excitement. And people have been coming up with ideas to use transformers, for example, now for our single-cell data in a multimodal fashion, even not just RNA, maybe ATAC, spatial, do some pre-training with these transformer-based settings. And then you get all these use cases. Quite a few papers coming out with this idea. At the moment, maybe not as competitive as people were hoping, but I think things are going in potentially in that direction. So the question I wanted to formulate here as my last thing is, can we formulate like this foundation model paradigm also in our single cell setting? As I said, there have been the transform-based ideas before. People have been just also making like a very large scale models. This is similarity from Heimbeck and Avi's lab. Maybe a way to go as well. Or this traditional thing that I showed you before, we basically learn an atlas and we transfer on top of this. This could be also a way to go. But I think it's an interesting direction, but it's not clear if we really need that big model capacity of these large-scale transformers. If you train some of these, a lot of compute goes in there, and often the actual results and benchmarks are not that encouraging yet. So maybe we're still in this scaling regime where we don't actually improve. Also, it's not clear, these are things typically made for sequence-to-sequence data. I mean, we don't have a sequence structure in gene expression, right? We do have one on sort of the genome, but not on predicted genes. So I think it's sort of vote is still out there. A lot of people comparing things, but I think it's an exciting proposition. In order to sort of give a bit more concrete tasks, one thing we've been doing is now to have a very simple, more sort of supervised particular case, in this case, cell type prediction. What we learned early on already is that organ-specific cell type prediction is kind of boring. There you need linear models only. So there have been our observation, many others as well, that linear models pretty much do the job versus neural networks. And for example, state-of-the-art cell type is just a logistic regression model, right? And that does very good annotation for particular organs. But if we go to organism-wide ones, where we have this strong ratio of cell types, maybe there we need some nonlinearity. And this was sort of our hypothesis. This is what Felix and David, who was PhD in the lab, is now a fellow at the Broad Institute, where we've been really trying to do human-wide cell type labeling. This ACTAB paper came out before. We've been taking cell by gene, something like 22 million cells, so really upscaling things, more than 5,000 donors. and a lot of shared annotation per tissue, something like 160 cell type labels, trying to predict cell type label. Okay, so this is what we try to do. We came up with a variation of a simple multi-layer perceptron, in this case using something called sort of tabular attention extending TabNet, which has like some type of transformer architecture that I won't explain in detail. But essentially, this sort of similar feature transformation type of ideas that you might know from transformers as well. We adapted this to the single cell setting, had a bit of an idea of an augmentation scheme, and I mean, the details are in the bioarchive that came out. And I should highlight, this is together with Evan and Chloe Villani from the Broad. The interesting thing is now, this is the behavior of a linear model versus our SETAB model for just a smaller number of samples. And initially, we don't outperform the linear model. But now comes the question, hey, with scaling, do we actually do more? And this is actually what's happening here. So we see that the linear ones sort of go up, but at some point flatten out with the number of cells, whereas this one sort of still keeps improving. And I think that's interesting. This is sort of where you can show that model capacity of this larger scale model is actually needed. We can zoom a bit more and see what's going on. And you can, for example, compare the cell types where I showed you the state of the art that gave you an overall AUC. In this case, we just sort of went to a smaller scale model of 0.73. Then if we train a linear one, sort of bit more larger scale data, what we did here, we improved. But then we see that simple XG boost, which is actually hard to beat, is not so well as the SETAB. So SETAB is sort of outperforming these things before, and you can zoom in and you can see how it performs for different organ types. I'm gonna skip this and sort of just conclude that the latent space here is interesting, and you can essentially also add an uncertainty score and have this model for scatter prediction. The last 4 minutes, if I may, I wanted to say how we now extend this versus the simpler prediction model that I was showing. And for this, I think adding space is an interesting aspect. I'm sure you've seen that spatial transcriptomics has been named Nature Method of the Year a few years ago, and a lot of these data sets becoming now commercially possible to do and available. I think it's getting an interesting time where you can potentially learn about interactions, modules, and so on. We have tools for that. Just came up with a formula called Spatial Data, together with Ollie's and Josh Moore's lab, where you now can actually superimpose multiple of these data sets and really deal robustly at scale with spatial data sets across different technologies. Check out that technique, check out SquidPy, how you can then actually implement this. I think it's a useful setting for multiple spatial line data sets that could have different origin and could be sort of combined and transformed together. What I want to ask now is, can we also formulate this foundation model idea in that spatial setting? And what you would observe, interestingly, is that if you take a cell and you isolate it, it has seen its neighbors. So it actually integrates information from the spatial niche. This has been well known. It sort of talks to its neighbors, and ideas such as cell phone to be based on the fact that there's sort of shared receptor ligand interactions, and you can sort of learn what type of communication happen. NovoSpark, one of these optimal transfer-based ideas, actually reconstructs information based on this pattern. And our NSAM graph neural network for this uses this information to sort of learn about local interactions. So this has been known before. The question we ask is, can we just take a disassociated cell and systematically learn and predict niche composition just by telling a cell, local cell? So can we take a disassociated single RNA-seq data set and predict its local niche? And I think that's an interesting task where you actually potentially need this large-scale data. So this is what we try to formulate in one of these foundational model settings. This is work by Ale and Anna, and this is really, so this is unpublished and sort of early times, so just one slide, but I really wonder what you think about this. Please give me feedback and push against this if you don't think it's useful. So we take one of these transformer-based approaches here, but train on large-scale disassociated as well as spatially disassociated samples, but then formulate spatial tasks such as predict me local niche density, predict me local niche compositions, and so on. So this is the downstream task. This is the self-supervised task. We call this niche former. Transformer of the local niche, and you can find interesting things. So in this case, for example, we look at Hongkui's mouse brain data set, huge one, and we can just ask, can we predict setup composition of the niche? And versus an MLP, so already nonlinear baseline, we quite strongly improve versus zero shot and in the fully local version. If we zoom in particular region here, I think in hippocampus, So this would be now actual space composition. We see here there's some blood vessels, I believe, if I look at color here. Here vascular cells as well as some other type of neurons up there. And then we can sort of locally predict the niche and we say, yeah, hey, here it's mostly vascular, right, in this region. And the other ones, you sort of find these GABA-associated niches and so on. So you can reconstruct some of these niches, and we're currently following up sort of more systematically, building these larger scale models. And we can see that they actually really outperform baselines quite significantly. So I think it's really interesting for spatial analysis in the future. So this question of could there be an underlying enabling base model? I think with these organism-wide data sets, we can train cross-tissue models to maybe predict cell type, but also niche composition at scale. So with this, I just have 3 more one-slide outlooks, if I may. I think in order to make all of this happen, it's really imperative, I think, to put some effort into the engineering side of things. Because if you want to do this at scale, there's a lot of just sort of hand things to be done. And early on, when we did our first singles analysis, we didn't do that, and it was like a mess. And first paper took us like 2 years or something, and then we came up with ScanPy to sort of make things faster. And since then, because it's not something you can maintain at scale, we've been coming together with a lot of labs, such as Stegle, Josef, Finotello, Wolf, Berger, Perigev, Teigman, and other labs to build what we call SCVerse. We now have this shared data structure, then a shared set of core packages that are all compatible and jointly maintained, as well as this ecosystem. So if you want to use single cell analysis, we check out this, maybe contribute your package there. I think it could be really one of these sort of Python-based versions of Bioconductor for these type of analysis. And we also came up with a best practice thing that we think is quite useful entry point, this living book online at SC-best practices if you want to learn about these approaches. So I think education and doing this hands-on thing, I think is really satisfying and useful. I said, I'm quite interesting about trajectories. One exciting method from actually originally more math and then machine learning is called optimal transfer, together with Dominic Giovanni and Marius, but in particular this optimal transport legend Marco Couturi. We've been coming up with what we call MOSCOT. We cannot do this optimal transport for time resolved data. I won't be explaining this in detail, but I think it's really a fun method to sort of concatenate now data across time steps, also spatial settings. And I really encourage you to check this out. The second thing, which is really very unusual for us, but we had more medical data coming up, so we're thinking, could we take some of these concepts and also expand to the phenotype domain? And if you look into medical annotation, particularly say from electronic health records, very often people just do hypothesis-based analysis. So not the unsupervised case, that now this idea of foundation models is sort of really invigorating. So with Lucas, we've been spending 2 or 3 years now working on, again, software framework at the back end to just enable these analysis that we kind of nicely can do in single cell, but we can't do on these phenotypic data because it's just a mess. So we want to do some type of exploratory analysis to really do big data things. And this framework is kind of transparent, sort of building upon your local EHR ecosystem, whatever you have. And we have a bunch of examples in the paper. Check it out. Just came out of mid-archive at Christmas. But essentially, you can take multimodal of these data sets, put them in a shared and data type of format. Then after some QCs and imputation, you can sort of build these patient landscapes as we commonly do for UWebs. And I really hope this thing catches on and could be a way how we deal now in patient relation also on a longer scale. The last point I'm trying to make is, I spoke a lot about these neural network architectures. I think we can take a lot of stuff out of the box, but in biology, we have all of these beautiful priors, all of this literature information. So can we build this more in? And one of the beautiful things I think we saw in AlphaFold is that they made not just any type of MLP, but they made the model group equivariant, so invariant on the rotation. That the thing is by construction like that, and then it generated things. And maybe we can also, in this quest for learning our regulatory networks, build in such information to then combine maybe our sort of prior-based things with our data-driven approaches, as Peter Sorger in this beautiful review pointed out. And some of these ideas happening. I said we do stuff for graph neural networks. Similar, we do this for gene programs, for RNA velocity together with the Josef lab, or for also spatial settings in image-based screens. So I think interesting times. And the field is, I think, really thinking about how some of these additions come. There's a lot of really in flux from machine learning in terms of these ideas. Let's see where this goes. So with this, let me summarize. I talked a bit about sort of state aggregation, building atlases and learning about these transitions. We then use this to build cellular organ atlases. We map new samples using transfer learning, some of these SCRs type of ideas. We predict response on top of these atlases using generative AI, simple latent space arithmetics, more complex settings. And we train and test some of these large-scale potential tissue foundation models. This is a lab at recent retreat. This is how beautiful Bavaria often is. So if you want to come by, let me know. And thank you again for your attention. Looking forward to some discussion.
Thank you so much, Fabian, for this really impressive array of all computational methods covering all problems in single-cell data. So we are open for questions. If anyone wants to ask, you can unmute yourself or you can type in chat. We have one question in chat. Regarding interpretability modeling drug responses, you seem to assume that drug responses are identical or similar for different cells, but what evidence grounds this assumption?
So this assumption of, I'm not sure that that's really the assumption. So maybe I wasn't entirely clear. So if I were to use a linear model in actual gene expression space, then you're right. Yeah, then the assumption would be that they're all sort of moving you in one direction. And we've been taking this idea from computer vision, where people try to I said this example sort of with the programming injector, but I think originally it was sort of putting glasses on people's faces. And all of our faces are a bit different. That's like a different ratio of eye to nose or something like that. So if you were to do this in pixel space, it would not work. Similarly here, gene expression space, yes. If we were to make that assumption, we would be back to essentially linear models, these factor models, these NMFs and so on. What people do, but which does not work in this complexity of the single cells because there's just too many nonlinearities there. So the key trick is that you go in this non-linearly trained latent space, where sort of this, which sort of, if you want, right? So you have sort of this really nonlinear thing that in latent space suddenly gets flat, and then there you do your linear thing, but then you sort of deconvolve it, you decode it back to the real space. That's why a linear superposition in latent space would actually turn out in a nonlinear effect in real space. So that's why SCGen essentially was working. And that's why also this extension of this sort of linear latent space model of CPA was doing good things. There have been quite a few of other approaches than based sort of on these original benchmarks that we did, such as Gears and others, where people have been then specifically formulating this as a prediction problem. But I think all of this is based on that in latent space, you have these sort of more simple type of models possible.
Okay, thank you. There's another question. Can you comment on the ability of current integration methods to take on cross-species cell type or perturbation comparisons?
Yeah, beautiful. I kind of skipped that. In the SCGen 2019 paper, we had already an example. There was a data set from I think Teichmann lab, where she's been looking at some type of immune response between mouse, rat, I think rabbit, and maybe human. And because it was sort of a similar perturbation, some type of immune, I think some interferon signaling or so, you could see how immune cells would be responding potentially similar or differently. And with SCGen, we could transfer between species at least a little bit that signature. So I think the idea of using this evo-devo type of techniques, at least this sort of comparative genomics techniques, have been around. People have been playing a bit with it, but I think it was also very preliminary. There have been some advances made. I think there's a beautiful paper using, I think, something like, it wasn't graph neural networks, it's just graph similarities from Leskovich lab. But I think also Marinka has been doing something in this direction from Boston, using essentially protein language models to really then embed transcripts across species. And there has been one of these sort of more foundation model ideas recently coming up again from Yuri, called UCE or something like that, where I think this is potentially useful. In the example that I showed, so our joint model, I think we go to up to now 100 million or so cells, which actually are taken from different species. And we essentially learn a shared set of orthodox as well as then disjoint ones, and you sort of adopt the embedding there. So I think we will in the future more easily be able to map things in a joint space across species, and then also compare cell tabulating and so on. I'm not sure how this would reflect the perturbation setting though. So yeah, I haven't really thought so much about this. So you're saying essentially that you might have similar set of like disease or perturbations across species you want to analyze. Maybe development would be the first type of perturbation that I would look at, but maybe Adam, if you want to explain more on that, happy to discuss later.
Awesome, thank you. Another question, what kind of physics-based models is the state-of-the-art for spatial transcriptomics? One is the POTS model. Are there any others? Can quantum models be used for spatial or single-cell genomics?
I'm not sure really. So, in the last one, last slide, this idea from Peter Sorger of combining sort of prior knowledge models with these data-driven ones, this kind of tries to add a bit of sort of more sort of forward-based modeling. I don't know really if anyone has been using something like a POTS model or an actual real growth model very robustly in these settings. So if you want to sort of take a step back and don't be fully spatial transcriptomics, one thing you can ask is, nowadays that we observe time series of single-cell RNA-seq data sets, can we just really just make a simple growth model or something like that? For cancer, for example, I think a key focus of I don't know, Elham, how much you focus on that, but I know Dana for the is a big thing, right? That this, let's say, growth of, for example, I think similar development, you could potentially combine with readouts. Not sure about that. And quantum model, I don't even know exactly what you mean SOP. Maybe you can explain that. I haven't seen that in that combination.
Hey, Devkanta, I don't know if you're here, you want to add any explanation? Or we can move to the next question. I wonder how ambitious you think we can be with foundation models. Ultimately, would we be working towards modeling perturbation effects at the gene or transcript or protein levels all at once? Is it data collection that makes this?
Man, this is I think what drives a lot of us, right? So I mean, are we all like working towards one GPT for single cell expression? Maybe, and some people really thinking that, are we working all towards one GPT of everything, and then you have the your particular single cell as a special case. I don't think we really believe in that, right? But I mean, some people hypothesize that. I do think quite strongly that some pre-trained embedding will be the way to go. In a sense, these atlases are something like these pre-trained embeddings, right? We have generated so many PBMCs that at least in the healthy setting, it's maybe not necessary to do that anymore, right? We just have enough of these samples. So maybe we just take a big embedding of PBMCs, such as the one that we have in SCPODI or some others, use these, and then only maybe have a bit of control to anchor our new sample, but then we just have this particular disease that we have in terms of PBMC and project on top of that. So I do think using references will become a standard. And in a sense, a foundation model is just a very fancy version of a reference, right? So that could be something going in this direction. How this will be across scales and how this will be across all the perturbations, I think it's still too much out there. And a lot of things will come. And I can say with quite some confidence, I think that the current models are not as exciting as they could be. If you look at the gene form, a lot of people playing around with this, not that useful yet. And I think just the normal SCVI, just a normal autoencoder type of embeddings often outperform these, maybe even sometimes linear models. But the promise is there, and other fields have been showing this. So I think maybe it's a matter of time and data, and unfortunately also compute.
Amazing. I think this is kind of related to that. Do you think just more data will help us to overcome the trade-off between integration performance and observation? I think you kind of touched on that.
No, yeah, but I think Darius is raising a point there, right? I mean, I did not. So yeah, that's actually an interesting point. So for these autoencoders, we kind of told the model that this is coming from different sources, right? In these more transformer-based settings, maybe just throw a bunch of samples at it and it learns it. So in this SE-tab, remember this was the cell type predictor across all of organisms. We did not tell the method whether things come from different samples. So it had to different patients or so. It had to learn this by itself. And this is kind of what you do in, let's say, ImageNet, right? Modern computer vision, you often take pre-trained ResNet or something like that, and you don't like learn the original filters by yourself. And those have not been trained by, it's saying, hey, this comes from that particular iPhone camera and this images come from that setting or so, but it sort of learns it by looking at many, many things. So I do think, actually, I'm quite convinced that looking at more data, we should enable a model to really discern technical artifacts from just the natural relation that it would see. Yes.
Great. Next question, what do you think about the generalizability of models built on single cell data, for example, limited number of patients, and is there a way to increase generalization?
Yeah. I mean that's kind of the idea of this foundation model thing, right? So if you throw many, originally we've been really afraid doing something across organs. We haven't even solved like doing a very good atlas for most of the organs that we have. I mean, we really did a lot of efforts with, I think, more than 50 people to jointly annotate the lung. And we don't have as good annotations for most of the other settings, right? So I think a lot of people have been hesitant to really just throw so many of these things together. But now people do it, even though annotation is kind of a mess and everyone, particularly immune cells setting calls things differently, but you're potentially getting this. So I think those show to help us generalize, and I briefly showed this organoid atlas, remember? So I think it's really interesting bridge between the completely in vitro setting of cell line and then our completely in vivo settings. So maybe, there's also a domain transfer translation type of thing happening that sort of tells you how this cell line would potentially then sort of look at this more tissue type of setting. So I really do hope generalizability comes up. But, we can now start measuring this. There's way how to quantify out of distribution behavior and so on. So I do think this is improving. And I showed scaling behavior for a very simple cell type prediction, and we really showed that this was improving there. So I really hope that more and more of these foundation model papers start coming up with scaling analysis. I think that's really important to show that in a simple setting, kind of on the linear performance, but then you at some point need to show that your model outperforms these simpler capacity type of problems.
Makes sense. Next question, you mentioned properties for different neural layers and architectures, and indeed gene expression is not sequence like text. What architectures do you believe would catch best the type of signal in this data type?
Man, this was really driving me nuts when I saw the first transformer papers. People saying like we literally have tabular data, right? I mean, that's what you would mostly assume with gene expression. And then you somehow do something in order to make it sequence based. I don't know if Ahmed, if you looked at the GeneFormer paper, this was Nature like midsummer last year. The trick they used there was after some filtering to essentially bit normalize and then rank the genes.
Mm-hmm.
And then from high to low, it looks, this is sort of the sequence that you have, and they do something. It's a bit strange.
Right.
If you think a bit more about what transformers do, though, they have this, essentially they have this long range attention, and it's actually not, in its original definition, if you remove the positional encoding, a transformer is actually a set-based function, right?
Mm-hmm.
The order doesn't matter. So it's like multiple instance learning in a sense. And then that would actually fit. So maybe it's sufficient to do just something tabular. And this is what we tried in this SE-tab. So in the SE-tab, we just have a tabular type of assumption, and it kind of worked. So I think the vote is still a bit out there. Also, if you look at SEGPT and other stuff, such as the one from Bo, which I think is exciting, some of them try to do reconstructions, some not. So I think people will still play around with the architectures a bit. Unfortunately, the playing around with architecture is so much more expensive because training is just really taking time. So yeah, let's see where the field converges. I don't think we know yet.
Totally agree. All right, maybe one last. What do you think is key to overcome technical batch effect from various labs and cell sources?
Yeah. I mean, yeah, as I kind of outlined now, I think throwing a lot of data at it could be a way out. Of course, throwing a lot of data at it and just sort of swamping everything with PBMCs might actually make things a bit more difficult if the capacity is not enough, because then you sort of lose the really small, interesting minor cell types that you might actually want to find. But in principle, I mean, we kind of see, right, that, GPT is quite good with very sort of small occurrence type of things. So I think if capacity is large enough, you should be able to deal with this. So yeah, that's a bit of the hope. But it's just one way. I mean, maybe linear, simple methods in many cases are just sufficient. So it's really important to have simpler baselines and not just always shoot with this big cannon of making everything transform, I guess.
Very reasonable. Right, one more question here. Do you think our prior biological and medical knowledge will be crucial to ultimately model a single cell causal ML? What kind of knowledge representation do you think is most promising?
Yeah. Yeah, it's like a back and forth. So in Germany, we actually have some bit more traditional institution that has been. So did you guys know, right? AI is an interesting term, right? I think de facto nowadays when we say AI, we kind of equate it with machine learning, right? But there's this whole set of AI that's not machine learning that uses like symbolic reasoning, knowledge representation and stuff. I mean, people start picking this up again to maybe make their textual inferences more informed. So yeah, I don't know. I mean, that's why we started looking into this therapy, right? So we've been starting to look into trying to aggregate a lot of now clinical information as sort of more complex phenotype than just cell type label, and to ask potentially harder questions. So I do think we need to work on the use cases. In the end, whatever we do methodologically, that's fun, that does not bring us some addition, maybe not that interesting. And I think many of these early transfer learning things, even us included with this SCGen, are kind of not yet solving really hard problems. So that's why maybe we need to find harder problems, and those problems typically based on prior knowledge. I mean, if you want to argue in this way, I think one of the key advances what made AlphaFold possible, of course, is this aggregation of questions and prior knowledge that people have been doing in these competitions. So maybe in the future we have a lot of competitions and then people coming up with things.
That's any other questions from the audience here? Maybe I'll end with one. I'm interested in your opinion about similar questions, but in the spatial world. You mentioned NCEM, and I wonder what kind of additional challenges you think we have to solve in integrating different types of spatial data. Some are single cell resolution, some are lower dimensional data sets, and different technological limitations. And in really kind of building models that can help us disentangle the niche from the location, from the composition of cell types, and getting insights into cell-cell communication. What do you think would be the most promising approaches there?
Yeah, thanks, Alam. I think it's really good to ground this a bit more in reality again, right? So we have all these beautiful spatial data sets coming up, and I don't think there's like this need for all this complex machine learning. I mean, I think, I don't know, maybe you disagree, but I have the impression we don't really know yet how we would compare a local spatial omics observations across patients. Like, what do we actually do if we see two microscopy images, kind of, right? I mean, they all look different because there's some shared architecture, but it's very different architecture. So we typically will burn it down to some simpler group statistics. We count number of neighboring cell types, some spatial correlations or so, which I think is good, right? I mean, we like spatial stats, I think is very useful to do for that. Later, we can come up with graph networks and compare stuff. But even though, in Human Cell Atlas already 6 years ago, we said we want to build a spatial atlas of all of these organs, I don't think we really know what that means. For disassociated RNA-seq, we have this beautiful metaphor of a cell type, which, gives us this way of aggregating things and then be able to compare. I think the similar notion for spatial is not entirely clear. I think it could be the niche. There's, again, also nice work from Dana's lab with just coming up with metrics to compare local clusters. And that could be a way to go. But yeah, I'm very interested to learn what you think here. I think vote is still out there.
Yeah, really looking forward to chatting more about this actually, maybe in the next meeting. I'm not sure if there are any additional questions. We're running out of time probably. No more questions? Well, thank you again, Fabian, for the fantastic talk. I think there are many appreciative comments on the chat as well. Really thankful for your time and impressive work.
Yeah, thanks for staying around and this really exciting discussion. It was really fun. Thank you.
Thank you, everyone.